students. So today let us start with the Neet discussion. One second, along the line. Neet discussion on topic cell, and let us start with the synopsis. Let us start with the synopsis here. So before going to that, the discussion, you should know what all topics are present. Highlight I will give you. That is, we have already studied the prokaryotes, from prokaryotes to the eukaryotes, and just the first topic. Recently we have studied the chapter cell, and in cell we have cell theory and introduction and modern cell theory, and also types of cells also we have studied with diagram, types of cells, and briefly we will discuss all these subtopics here, and differences between prokaryotes as well as eukaryotes. Prokaryotes as well as eukaryotes differences, and after that plant cell as well as animal cell. Plant cell as well as the animal cell. After differences, one more important, that is diagram, plant cell as well as the animal cell. In prokaryotes, so we have cell wall discussion we are going to do. Cell wall, then cell membrane. I hope you remember all these topics: cell wall, cell membrane, structure of a cell wall also we can see here, and after that cell organelles, which includes plastids, chloroplast, plastid, chloroplast, mitochondria, ribosomes. Okay, mitochondria, ribosomes. Then detailed study of the chromosomes also. endoplasmic reticulum, Golgi complex, lysosomes. So apart from this, we also have the centrioles. If possible, we will discuss all the things. Centrioles, okay, next. So let us complete all these synopsis one by one. So what synopsis we have? So overall in cell we are having cell theory we are going to see. Before that discussion about the history of the cell, also we will see some scientists who discovered about this. After that, types of cells. Orally, we will discuss all this introduction part, and some which is not given in your NCERT. Just go on writing that information on the board. And prokaryotes, eukaryotes, almost you are very much familiar. If you are not familiar, you have to remember differences between prokaryotes as well as eukaryotes. So this and this will be important for your theory, and cell theory also. You may get objective questions or you may get in the theory also. And cell wall, cell membrane, that is fluid mosaic model. I hope you remember. Okay, fluid mosaic model you are going to study in cell membrane and a cell wall, and after that plastids, chloroplast, mitochondria, ribosome, Golgi apparatus, centrioles, microbodies, and these are all different types of organelles which are present in the cells. Okay, so very much important, and most probably everything is present for your test, so we can discuss with the history. So to discuss about the history, so as you know, the cell is a structural and functional unit of living organisms. Cell is very important and it is one of the characteristic of all the living organism. Yes, so it is a very important living organisms. And if you talk about the scientists who discovered about this, they are all, that is the first cell means you will be coming to know about the Robert Hooke, right? Robert Hooke, he was in 1665, Robert Hooke, he has discovered about the cell, and he described cell is micrographia. So what he described is the cell is considered to be as a micrographia. Micrographia. Next, after Robert Hooke, so next Leeuwenhoek is one more scientist who observed animal cell, especially who observed the animal cell. He was the Leeuwenhoek, and he used the term means like Robert Hooke used the cell as a micrographia. Same way, so Leeuwenhoek used the term as in animal cell, that is animalcules. Because during that time it was not called as cell. Different scientists has given different names. Okay. Now after this, So the study of this structure, cell, what do you call the study of the cell is called as what? Cell cytology. Okay, study of tissues, histology, right? So here the study of cell is called cytology, and this is given by the scientist Animal Hughes. After that, Robert Hooke is considered to be as the father of cytology. Who is the father of cytology? Robert Hooke. In 1665, he was the first person to observe the cell. So he is considered to be as the father of what? Cytology. And one more scientist, now father of modern. He is old. We are going to consider Robert Hooke as a old. So who is the modern cytology? That is Swanson. You should know all these things. The modern father of cytology is Swanson. Next, coming to the cell theory. Okay, let us discuss about almost you know that cell theory. Matthias Calden, okay, he is a German botanist, and one more scientist, Theodor. Okay, one is Matthias and one more is what? That is Theodor Schwann, who are proposed this in the year 1839. Both of them has explained about the cell theory. So in this process, we are going to study all what they included, what they explained about the cell theory, Matthias, and one more scientist, Theodor Schwann. So main feature, all organisms consist of cell. Yes, first time they told almost all living organisms are made. Nowadays we know all cells are, but during this 18th century it was not known. So what they discovered, both the scientists, that is all the cells structurally, metabolically similar. All living organisms are made up of cell. Structurally, metabolically, they are same, and also they perform all the activity of the organism. Yes, because each and every way our metabolism is depending upon the cell, and each cell is a heredity also. Okay, it can be hereditary. Next one. So next, modern cell theory, the cell can survive independently, but organelles cannot survive independently. After cell theory, apart from textbook and all, so we have modern cell theory also. So modern cell theory, it is telling, that is all living organisms are made up of cytosol, nucleus, organelles, right? And the function of this activity are all depend upon the cells. So cells can live independent, but organelles which are present inside the cell, they cannot be living. Next. So coming to the types of cells. Three types of cells they have differentiated. Undifferentiated cells, differentiated cells, then dedifferentiated cells. Okay, normally types of cells are three types: differentiated, okay, undifferentiated, as well as dedifferentiated. So what do you mean by differentiation? So here undifferentiation means stem cells. Example, here we can give stem cells. Okay. Stem cell. They are specialized. They are having the power of divisions, and root and shoot, skin cells, germinal epithelium, bone marrow, zygote, etc. All these comes under undifferentiated cell. Whereas differentiated cells, also called as post-mitotic cells. Okay. So after mitotic mixed post, they are specialized to perform the function of the cell. They are specialized for the function, and cell division is absent in case of differentiation. De-differentiation. Healing of wounds. If any injuries takes place, okay, the rupturing or some wound will be taking place on the skin. So that wound will be, after some time, that wound will be what? Cured. Why? Because of de-differentiation, new cells will be formed. That new cells will be formed. Next, coming to the cell envelope. So as you are very much familiar with the cell envelope and all, so which is consisting of the different and also cells types. We have completed cell types, means we have RBC, WBC, okay. Then apart from that, columnar epithelial cells, we have neural cells are also, that is neurons are present, tracheates are present, xylem are present, eukaryotic, that is animal cell is present. So different types of cells, bacterial cells are present, etc. Okay. So next, coming to the differences between prokaryotes as well as the eukaryotes, if you see. So prokaryotes and differences between eukaryotes, orally we can tell. So what will be the size of the prokaryotes? The size of the prokaryotes will be 0.1 to 5 millimicron. Okay, it is the size. Whereas in case of eukaryotes, it will be 5 to 100 millimicron. Okay, prokaryote size and eukaryote size. Next character, prokaryote cell has one envelope. Prokaryote is having only one envelope. But in case of eukaryote, if you take, there are two envelopes. Okay, there are two envelopes. In case of, like, plasma membrane also is there, cell wall is also present, right? So eukaryotic is having two cell membrane. So nucleus is not very important. Prokaryotes, they do not have well defined nucleus. So but in case of eukaryotes, well defined nucleus is present, having the chromatin, okay, that is fibers, nucleolus is present. Cell wall, if present in prokaryotes, it is called as muramic acid. Okay, cell wall is present also, it is called as muramic acid. In case of eukaryotes, if you take, the cell wall is, if present, muramic acid is absent. Here, cell wall is called muramic, the cell wall is not, that is muramic is absent, acid is absent. DNA is naked, it means histone is absent. Here DNA is present, it means histone protein is present. Okay. And next one, DNA lies freely in the cytoplasm. In case of telling the differences, DNA lies free in the cytoplasm, whereas in eukaryotes, so DNA is present inside the nuclear membrane. Okay, they are not freely suspended in the, and some DNA is also present in the plastids. In the plastids and in mitochondria also, the DNA, different organelles are also having the DNAs. Okay, next differences. Here in case of prokaryotes, DNA is circular, you know that. Okay, they have circular, if DNA is present, they have circular DNA. So here DNA is linear. Okay, the DNA is linear. And next one, and extranuclear, if it is present inside the mitochondria, it will be circular. Linear also is there, circular are also present in case of eukaryotes. Next differences, if you see prokaryotes, cytoplasm does not show cyclosis. Okay, cytoplasm is not showing any cyclosis. Cytoplasm is showing the cyclosis in case of eukaryotes. Next differences. Membrane bound organelles like mitochondria, Golgi body are absent in prokaryotes. All the organelles which I have written here, all will comes under eukaryotes. Got it? And next, gametes are formed in case of prokaryotes. So, gametes are formed since sexual reproduction meiosis are absent. Gametes are formed, but there is no sexual reproduction and there is no meiosis in case of prokaryotes. But in case of eukaryotes, gametes are formed, sexual reproduction is taking place, as well as what? As well as the meiosis is also occurring. Microtubules, microfilaments are absent, very important. Microtubules, filaments are present in case of eukaryotes. So spindle fiber is not having any division. Spindle fiber are not present here. Spindle apparatus, nuclear division during that time. During mitosis, we will be having centrioles. So with that, spindle fiber formation is helpful. Okay, that is absent here. There it is present. Next, 70S type of ribosome is present in case of prokaryotes and 80S type of ribosomes are present in you. Fix it in your mind. Okay, 70S type of ribosome is present in prokaryote and 80S ribosomes is present in case of the 80S and 70S type in mitochondria and all. It is 70S type. Centriole is also absent in case of prokaryotes. Centrioles is present in case of the eukaryotes. Okay. So example, bacteria, mycoplasma, PPLO, plasmon, that is pneumonia, like organism. Okay, cyanobacteria. Whereas in case of eukaryotes, if you take, plant cell will come. Okay, animal cell will come, fungi will come, protista also will come. So this is for a very important differences between prokaryote and eukaryote for theory as well as for the name. Most probably they may ask you 5 marks question also for theory. Next, plant cell, animal cell differences. Again, this is also expected question. If the paper is easy, they will be asking this type of very easy. What you will do, you will be just neglecting the differences. You have to go through and minimum you have to write 8 to 10 differences to get 5 marks. Okay. So let us discuss about the orally. We will discuss plant cell, animal cell differences. Okay. So almost you know that the plant cell. First we will take it. The plant cell is having rigid cell wall. Okay, the cell wall is very rigid. Okay, here cell wall is absent, you know that. Okay, after that, plastids are present in case of plant. The plastids are absent in case of the cell wall, and the plastids are present in plant but absent in the animal. Next, after this, the mature cells has large vacuole. Okay, the mature cell have a large vacuole, but in case of animal cell, the vacuoles are what? They may have very small vacuoles, not a large vacuole is present. Nucleus lies at one side in the periphery, but in case of animal cell, if you take, the nucleus lies exactly at the center. Okay, they will lie at the center. Then, after this, the plant cells, centrioles are usually absent. Every time you remember, centrioles in the animal cell, centrioles will be present. But in case of plant cell, centrioles are absent. Here, centrioles are present. That is one of the major differences. Okay, almost plant cell, animal cell are very much familiar. You have to write, you should know cell wall, plastids, centriole is present in animal cell but absent in plant cell. Okay, such differences you should know. And after that, Golgi apparatus, number of distinct unconnected units called diatosomes are present. So here they are localized and connected with single complex. Reserve food material in case of plant cell is starch and fat. The reserve food material in case of the animal, it is usually glycogen and fat. Okay, starch, fat, glycogen, okay, glycogen and fat you can tell. Cytokinesis. During the mitosis, the last step where the cytoplasm division will be taking place. Cytokinesis means what? Cyto means a cytoplasm. Kinesis is nothing but what? Division. Division of the cytoplasm in case of plant cell, there it occurs cell plate. Okay, what the cell plate is so that both the cytoplasm will divide. Okay, that is in case of plant cell. What about in case of animal cell? So, in case of animal cell, the cytokinesis takes place with the help of cleavage. Here cell plate, here cleavage will take place. Okay. So, these are some differences. Next. Let us start with the orally prokaryotic bacteria. If you take, okay, bacteria, the structure, okay, the structure which you have already known before going to cell wall. The prokaryotic example we are taking what? Bacteria. So here bacteria which are present. So these bacteria in prokaryotes only, just orally what all parts are present in prokaryotes. Let us see that. So, cell membrane are present, cell wall formation, DNA replication, respiration, secretion, all these takes place normally in case of bacteria by only one cell, that is mesosome is present, which is absent in case of the, which are absent in case of the other organelles. Next. So apart from that, in cyanobacteria, the extension into the cytoplasm, we are talking about the prokaryotes cyanobacteria. So cyanobacteria, how it is present? In case of cytoplasm, chromatophores that contains pigments are present. Okay, cyanobacteria, chromatophores, you should know. The cytoplasm of the cyanobacteria consists of the which pigment, which is called as chromatophores. Okay. Next. After this, cell wall will be in the form of filamentous. Okay, flagella, the filament, the thread-like structure has divided into three parts. What are they? That is filament, hook, basal, and body helps in locomotion. Okay, filament, hook, basal, and also basal body and helps in locomotion. Besides flagella, we have pili, fimbriae. Okay, apart from flagella, we have pili as well as the fimbriae. And they are helpful for what? They are helpful for locomotion, moving for the bacteria. Okay. And pili means they are tubular structure made up of protein. Fimbriae are the small blister-like sprouting. The bacteria will be having a small sprout-like structure which are called as fimbriae. Okay, which are called as fimbriae. You know very much theory, NCERT questions. Fimbriae apart from flagella, bacteria also having the pili, okay, as well as the fimbriae. And 70 years type of ribosomes are present in case of bacteria, that is in the prokaryote. Reserve food materials will be present and stored in the cytoplasm in the form of inclusion bodies. So this term you should know. What do you mean by inclusion body? So prokaryotes, they will be storing the food. They will be storing the food in case of prokaryotes, where they are storing in cytoplasm in the form of granules, gandugal. Okay, that granules we will call it as inclusion bodies. What it is called as inclusion bodies. Next, after this, so you should know that some questions you should know. Virus. Okay, virus is actually a cellular, right? It is connecting link between the dead and the living actually. After that, the next smallest cell mycoplasma ppm we have studied. Largest unicellular eukaryotic. Okay, they are asking largest unicellular. Okay, unicellular eukaryotic cell Acetabularia algae. What it is? Acetabularia algae is a largest unicellular eukaryotic. Longest human nerve. Okay. Longest plant cell, that is rami jute fiber. Okay, jute fiber also considered to be as a what? Longest. Next, let us go for the cell wall. Next, coming to the cell wall. So, in case of the cell wall, we have seen that this was first studied by the Robert Hooke again. Okay, the cell wall was first studied by the Robert Hooke in the Koch cells. Where? In the Koch cells. And that is secreted by the cytoplasm. Okay, that is secreted by the cell wall is having, why cell wall is present? To protect the cells from the mechanical damage. Okay, to protecting the cell from the mechanical damage. And so the plant cell having the here the plant, it is found in plant cell wall in plant, it is made up of so in case of cell wall in plant, it is what? It is cellulose. In case of fungi, it is chitin. In case of prokaryotes, okay, in case of prokaryotes, like bacteria if you take, cyanobacteria if you take, the cell wall will be having, what type of cell wall they will be having? That is peptidoglycan. Peptidoglycan. In protista if you take, the cell wall is absent. The cell wall is absent in protista. Bacteria, cyanobacteria is having the cell wall, peptidoglycan, whereas plant cell wall is made up of cellulose. Cell wall of the fungi is made up of what? Chitin. Okay, you should know this. Next, it is absent. It is absent in animals and mycoplasma. Cyanobacteria and few protista also it is made up of peptidoglycan but absent in animal cell. Cell wall cannot be seen in animal and even mycoplasma also absent. In case of mycoplasma, it is absent. Structure of a cell wall. So, structure of cell wall 0.1. Okay, it is 0.1 to 10 millimicron. The plant cell having cellulose, bacteria and composed of peptidoglycan as well as DAP. It consists in fungi, it consists in fungus, it is chitin. In plant cell, it is cellulose. In case of bacteria, cyanobacteria, protista, it is peptidoglycan. Okay, the question may get from this. And chemical composition, cell wall is made up of what? Okay, the chemical composition of the cell wall, if you take, the chemical composition is, means cell wall is made up of what type of chemicals? First thing, water. Without water, nothing, the organelles cannot be formed. So water is having 60%, and apart from hemicellulose is 5 to 15%. Orally I am telling, note down. And pectic substance is present. After that, lipid is present, protein is present. 2% of protein, lipid is 0.5%. Okay, and pectic substance 2%, hemicellulose 5%, 60% is water. Okay, cell wall is made up of water, hemicellulose, pectic, then we have proteins and lipids. All these will form the cell wall. Cell wall are having microfibrils on their body. Okay, fibrils are also present. So fibrils and cellulose is the main component of the cell wall again and again. Cell wall is formed of following layers. Cell wall is having layers also. What are that layers? The layers are don't think cell wall is present only in plant. They are present in where? They are also present in fungi. They are also present in protista and also bacteria, cyanobacteria. Okay. Now, these are having what type of layers? These are having made up of layer middle, lamella, primary wall, secondary wall, tertiary wall. Okay, so somewhat cell wall structure if you take, it will be in this way. The ultrastructure So if you take the section of the cell wall, you can see this type of layer. So outer layer what you are seeing, this is a middle lamella. Okay, middle lamellae and this is a primary wall and these are all secondary walls, which is denoted as S1, S2, S3. Okay, and middle the space which is present is called lumen. So what are the layers of the cell wall? The layers of cell wall is having middle lamellae, primary wall, secondary wall moving towards internal, and at the middle the space is called as what? Lumen. Tertiary wall as well as the lumen. Next. So if you see middle lamellae having two adjacent, means middle lamellae is adjacent layer to the next cell. They are connecting one cell to the another cell wall, and these composed of the middle lamellae composed of pectin. Okay, pectin and calcium and magnesium pectate. It is made up of what? Magnesium, calcium as well as pectin. And if it is absent on the outer free surface, they are having plasmodesmata. Why? What is the function of plasmodesmata on this layer? So, whatever the circulation, water is there, nutrients, they will be moving from one cell to another cell. They will be having interaction from one cell to another cell with the help of plasmodesmata. Okay, plasmodesmata connecting one cell to the another cell, adjacent cell. Okay, next, after this, so primary wall, if you take, it is elastic. Okay, it is elastic, single layer. Okay, its diameter will be 0.1 to what? 3 microcellulose, hemicellulose is primary wall. It is made up of what? Cellulose as well as the hemicellulose. Pectin is also present. Next, whereas secondary wall in NCERT, some sort you have studied, in some you have not studied. Secondary wall, if you take. So secondary wall, it is diameter 3 to 10 millimicron in a diameter. Okay, and these are having three layers: S1, S2, S3. S1, S2, S3 is present, and these are having made up of, secondary wall is made up of lignin, pectin, lignin, suberin, cutin. Okay, lignin, what? Suberin and cutin are present. Means each layer is made up of, this is made up of calcium, magnesium and all. Here it is made up of what? These are primary is made up of the cellulose, hemicellulose and pectin. But in case of secondary wall, these are made up of suberin, lignin. Okay, after that cutin. Next. So cell wall thickening also will take place, which we are not going to discuss here. Cell wall thickening we are not going to discuss next. Coming to the next, next coming to the cell membrane. After cell wall, next coming to the cell membrane. What you have studied in the cell membrane? The cell membrane is elastic. Okay. These are usually present living, pliable. Elastic means what? Pliable. It can be mold. It is not stiff or rigid. Okay, it is flexible. Living, pliable, hydrophilic and selectively permeable membrane. Okay, it is allowing the ions only if necessary. So it is selectively permeable membrane. Then after this, coming to the structure of a cell membrane, how the structure is present, let us see. So structure of these are microscopic and they are having tri-layers, and chemically cell membrane is made up of, okay, chemically what it is made up of? Proteins, lipids, carbohydrates, sugar. Okay, so proteins will be 44 to 76%. Understood? And in case of lipids, 20 to 53% cell membrane is made up of protein, lipids, carbohydrate just 1 to 5%. Carbohydrate, that is 1 to 5%. Water will be 20%. After that, DNA will be there, RNA are absent in case of cell membrane because it is covering. Okay. Okay, next coming to the 52% of, in case of human, if you take, in case of plant, protein, carbohydrate, lipids were present. In case of percentage, if you see, animal cell, that is especially human cell, if you talk about cell membrane, it is made up of what? Proteins. Proteins, majority, 52% of cell membrane in human, it is made up of protein. Okay, 52% protein is present, and one more, that is 40% of lipid is there in case of human. Next, proteins are present, enzyme, permease, receptors are present in cell membrane. Okay. Next, lipids are usually phospholipids. What type of lipids are present? So lipids are present in case of phospholipids. 55% of total phospholipids. They are present in the phospholipids. Polar hydrophilic head is there in this way. Lipids are having phospholipids, head, tail. Head is called as what? Head is called as a polar hydrophilic. Hydro means they are accepting the water, and these tail, if you take, they are non-polar hydrophobic. They cannot face towards the water. Head is facing towards the water. So these are polar, these are non-polar. These are hydrophilic, these are called hydrophobic. Okay. Now, apart from this, carbohydrates if you take, usually what we have told, carbohydrate is also present in cell membrane. Oligosaccharides, the plasma in cell membrane, it is called oligosaccharide. What it is called, carbohydrate is present in the chains of oligosaccharides, okay, and form glycolipids and glycoproteins are also present respectively. Next. Cell membrane, they are arranged, fluid mosaic model if you see. In case of cell membrane, fluid mosaic model. I hope you remember, and these are very important for your exams and for objective also. Okay, cell membrane in that fluid mosaic model. Who proposed fluid mosaic model which comes under cell membrane? That is Singer and Nicholson. When? In 1972. When? In 1972, Singer and Nicholson has discovered about the fluid mosaic model. Okay, what is this fluid mosaic model? So, fluid mosaic model is plasma membrane, okay, that is containing lipid bilayer. What they discovered, lipid layer is there. That lipid is made up of two layers which are called bilayer. What it is called, made up of bilayers, okay, and in which proteins are also present, outer, middle, everything. Okay, next. Lipid if you take, they are made up of phospholipid. Lipid in case of cell membrane, they are made up of phospholipids, and they provide the particular term fluidity. They maintain fluidity. Okay. So after the discussion of fluid mosaic model, half an hour we will discuss the question. Rest half part we will discuss later. Next one, in case of lipid, it is maintaining the lipid provide elasticity. We are talking about the cell membrane fluid mosaic model. Okay, lipid elasticity, stability to the plasma membrane. Okay, next. So the fluid nature of the membrane is also important. Proteins if you take, so there are two types of protein, extrinsic protein as well as intrinsic protein. Okay, proteins means the cell membrane is also made up of protein. Extrinsic protein means what? So the protein which is present at the periphery. Okay, peripheral side. Periphery andre side ಹೊರಗಡೆ. Peripheral, 30% of total protein is peripheral. And in the intrinsic protein, some proteins are present, they are present where? Inside. Okay, these cannot be separated easily. 70% of protein is intrinsic. That is cytochrome oxidase. It is made up of cytochrome oxidase. 14 proteins are present. And these are integral protein are carrying what? They are carrying the permease, enzymes, receptors. They function as permease, enzymes as well as receptors. And next, tunnel proteins are also present. Transmembrane proteins are also present in the intrinsic protein. The The plasma membrane, if you take, they are oligosaccharide and asymmetrical. The plasma membrane are oligosaccharide and they are having form glycolipids from the and glycoproteins. Lipids, proteins are also present here. So both glycolipids and glycoproteins will form bond. The question is, the glycolipids and glycoproteins will form glycocalyx. What it is going to form? Glycocalyx. What it is called? Glycocalyx. Okay. Next, it acts as a recognition center. And entire, if you take the flock movement, transverse diffusions, everything, the plasma membrane, glycolipids, oligosaccharide part of the glycolipid, you have the diagram in textbook in this way. Here intrinsic proteins are present internally. Okay, internally these are the intrinsic. Peripheral on the surface some proteins are present. These are extrinsic or integral and these are integral. Integral proteins. 70% of protein are integral. Next. Let us see special point about the cell membrane. Okay. Some more points we will discuss about the cell membrane. So what points we are going to discuss here? That is, the electron spin resonance flip-flop mechanism is absent in protein. Protein and lipid undergo rotational lateral diffusion in membrane, and eukaryotic plasma membrane consists of eukaryotic plasma membrane cholesterol, but in prokaryotes hapenoids are present, no cholesterol. Okay. After that, porins, the intrinsic protein found outer mitochondria and bacterial membrane. Okay, permeates translocase carries the transport of material. Cell membrane is acting as differential permeable. Glycoprotein, glycolipids provide antigen, RBC antigen, fertilizing, anti-fertilizing reaction. So, glycoprotein as well as glycolipids, they also act as antigens and fertilization process. We need one chemical that is called what? Fertilizing and anti-fertilizing reaction. For that, it is going to be helpful. So, it will maintain, lipids provide fluidity, elasticity, stability, and also lipids which are present. These are the lipids. They are helpful for fluidity, stability, elasticity, and growth, cell junction, cell division. Fat soluble substance is directly entering into across the lipid layer. Okay, then water may cross the membrane from higher to lower concentration. Water will be moving from the higher to lower concentration, and this process is called osmosis. Okay, some lipid bilayer, non-polar lipid, they require carrier proteins. We are having, because these are not facing towards the water. So for transport of water, they need proteins. Okay, they need the proteins so across the membrane. So this are called facilitated diffusion. Some ions or molecules are transported across the concentration gradient by using ATP. Means we have active, passive. Passive, there is no requirement of energy. But passive. Active, in case of active, sodium potassium pump, which are moving against the concentration gradient, right? They will be affected. So they are having sodium potassium pump. Next. So let us discuss the questions. So the rest synopsis we will continue in the maybe in the next class. But now we will start with the question session. Okay, let us start with the first question. Okay, let us start. So what we have discussed in the synopsis. In case of synopsis, we have discussed about cell theory, history, types of cells, okay, prokaryotes, eukaryotes, plant cell, animal cell, cell wall and cell membrane we have discussed, right? Next we will start with the question. And which of the following statement is not correct? Okay, some topics we have not yet discussed, but it is completed in theory. Try to answer the questions. Okay, or else next session we will start with the plastics and all. Okay, but now we will solve some questions. Okay. Which of the following statement is not correct? Okay, so which is not correct they are asking. Lysosomes are formed by the process of packaging. Okay, packaging in the endoplasmic reticulum. So it's not correct. Instead of lysosome, ribosome should come. So I think first answer is correct. Okay. So lysosomes have numerous enzymes, that is also true. Hydrolytic enzymes of lysosomes are having acidic pH. Lysosomes are the membrane bound structure, yes. So answer for first one is A. Okay. Then the second one. In cell theory, we will come across Matthias and Theodore. They have told the concept of omnis cellula e cellula regarding cell division was first proposed by. Out of both of them, one scientist has proposed this, that is B. Rudolf Virchow, along with Theodore, along with Swan. One more scientist has also given, that is Rudolf Virchow. What he has told is Rudolf Virchow, the concept of omnis cellula e cellulae. They have studied here. Okay. Next, coming to the next, which of the following statement of mitochondria is incorrect? Okay, so they are asking about the mitochondria, which is incorrect. Mitochondrial matrix contains single circular DNA molecule and ribosome. Yes. Okay. In cytoplasm, the DNA will be present in linear. But that same DNA, if it is present in mitochondria, they will be circular. Outer membrane is permeable to monomers and carbohydrate. So third one, enzymes of electron transport are embedded in outer membrane. So that is wrong. Inner membrane is convoluted with infolding. Yes, infolding. Mitochondria are having finger-like projections, so infolding are present. Third answer will be C. Fourth, the shorter and longer arm of the submetacentric, we are talking about chromosome. Some will be having smaller arm, some will be having the, so in case of longer arms of the submetacentric, so it is P arm and Q arm. This is not given in your NCERT, you should know. Small, longer arms of the substrate, shorter and longer. Shorter are called as P arm and longer arms are called as Q arm. Fifth question. What is the pair of organelle does not contain DNA? Okay, that is nuclei are also there, mitochondria is there, and lysosome is also there, and chloroplast and vacuoles are also having the pair of or does not contain DNA. They are asking lysosome and vacuoles. Okay, but in case of B, pair they are asking, lysosome don't have, but mitochondria is having. So we cannot take that as a answer. The fifth one is D. Match the column 1 with column 2 here. Okay, match the column 1 and column 2. So in case of column 1, Golgi apparatus. Okay, in case of this Golgi apparatus they are asking. Golgi apparatus is nothing but what? What is the function they are asking here? So sixth one, formation of glycoproteins and glycolipids is the function of the Golgi apparatus. Next. B. Lysosome is nothing but digesting. Yes, like always remember, lysosomes are indirectly they are responsible for digestion. Next, vacuoles. Vacuoles removal of the waste, excretory product. They are linked to the excretory products. Next, D. Ribosomes. Ribosomes synthesis of protein. You know that. Okay, ribosomes main function is the factory of the protein synthesis. Okay, so answer for sixth one is 1. Seventh, which of the following is true for nucleolus? Nucleolus location is where? In the nucleus. So here it is the site. Always one point I'll tell you. Ribosome is the factory of protein. Same way, nucleolus is the factory of ribosome. Okay, ribosomes are prepared where? In the nucleolus. So answer is what? That is D. Where we are? That is the seventh one is D. It is the site for active ribosomal RNA synthesis. Always remember. Especially in that rRNA. rRNA synthesis is taking place where? In case of rRNA, that is in the ribosome, in nucleolus. The Golgi complex participates in, okay, formation of secretory vesicles is the answer. Which of the following events does not occur in rough endoplasmic reticulum? That is phospholipid synthesis. What it is? Phospholipid synthesis. 10th question, lamp brush chromosome. So 10th, that is lamp brush chromosomes is nothing but diplotin bivalent. No, lamp brush chromosome actually you are going to come across the in some amphibians and all these chromosomes will be present. So oocytes of amphibians. So answer collagen chromosome. Oocyte of amphibian is correct. Allosomes are sex chromosomes. Select those which are incorrect. Allosomes are sex, that is correct. Submetacentric chromosomes, L-shaped chromosomes, polytene chromosome, oocytes of amphibian. Actually, lamp brush chromosomes are oocytes of amphibians. Answer for 10th is D. 11. Which of the following cell organelles is responsible for extracting energy from carbohydrate to form ATP? Which is that answer? Mitochondria. Okay, which is the energy production will take place. Next, select the mismatch. Which is mismatching? Gas vacuoles green bacteria. Okay, gas vacuole green bacteria. No, it is not mismatching. Large central vacuole is present in animal cell, not in animal cell. It is present in plant cell. So B is the correct answer, which is mismatching. Protista is eukaryote, correct. Methanogens is a prokaryote, that is also correct. Select the correct statement. So 13th question we are discussing here. 13th question which is wrong statement. Let us see. Pylifimbrae, B1. The pylifimbrae are mainly involved in the motility of bacteria. So pyli as well as fimbrae are not present. Cell organelles. In case of bacteria, flagella act as a motility, not the fimbrae and not this pyli. A cell organelle containing hydrolytic enzymes, which is that lysosome. Mitochondria and chloroplast are what? 15th question we are discussing. 15th question, mitochondria, that is A is true, mitochondria and chloroplast, semi-autonomous organelles, but B is false. Division pre-existing organelle and they contain DNA but lack protein synthesis. Okay, so they are wrong. B is false, A is true. 15th is A. 16th, if you take your 16th, microtubules are the constituent of spindle fibers, centrioles and cilia. Okay, B answer. 17th, which of the following cell organelle is enclosed by single membrane? Okay, which is having only single membrane. Nuclei is not coming. Mitochondria, lysosome. 17th answer is A. 18th, water soluble pigments found in plant cell vacuoles are, so they are anthocyanin. They are what? Anthocyanin. Match the column. Thylakoids. Thylakoids means they are plant membrane, coin-like structure. Cristae. Cristae means infolding of mitochondria. They are called cristae. Cisternae. Cisternae are the disc-shaped sacs of the Golgi apparatus. Chromatin, if you take condensed structure of the DNA, are called chromatin. Answer for the 19th question is D. 20. Which of the following structure is not found in prokaryotic cell? Okay, it is not found in the prokaryotic cell, that is C. Nuclear envelope is absent in prokaryote. Cellular organelles with membranes are. Okay, cellular organelles with membrane are. Lysosome, Golgi apparatus and mitochondria have cellular organelles and also membranes. Okay, B answer. 22. Which of the following are not membrane bound organelles? Okay, which is not membrane bound organelles? That is answer for 22 is D. Okay, D, ribosome. DNA is not present in nucleus. It is having DNA. Mitochondria is having DNA. Chloroplast is also having DNA, but ribosome is not having. The 23rd answer will be D. 24th. A nuclear envelope is a derivative. Nuclear envelope is a derivative of the rough endoplasmic reticulum. The structures that are formed by the stacked organized flattened membrane sacs. Stacking organized flattened membrane sacs in the chloroplast are 25th will be, that is grana. The coin-like structure which are present in the chloroplast, they are having the grana. Next, select the correct matching in the following ways. So 26th question, rough endoplasmic reticulum, synthesis of glycogen, wrong. Endoplasmic oxidation of fatty acids, smooth oxidation of phospholipid. Smooth endoplasmic reticulum is synthesize the lipids. Answer for 26 is D. Next, 27. The chromosomes in which centromere is situated close to one end. Close to one end, that is 27th answer will be the acrocentric. Which one of the following is not an inclusion body? Found in prokaryotes. It is not found an inclusion. Glycogen granule is inclusion body. Polysome, actually we can take 28th D. Okay, 28th D. Rest, phosphate granule, sinophysin granule are all present. Next. The solid linear cytoskeleton element having the diameter 6 nanometer and made up of single type of monomer are known as what? Microfilament. B. 29th question. Answer will be B. Next. 30. The osmotic expansion of cell kept. Okay, kelp in water is chiefly regulated by vacuoles. Okay. Match the following. 31 question we are discussing. 31. Centriole. Centriole means basal body of the cilia. So basal body of the cilia. Chlorophyll, that is thylakoids. Cristae. Again repeating cristae, enfolding of mitochondria. Ribozymes, which are nucleic acids. Ribosomes are what? DNA is what? They are the nucleic acids. So answer for 31 is A. Next, 32. The Golgi complex play major role in translation of protein and glycosylation of lipids. Golgi complex, what it will do? It will translate, modify, pack and the proteins and glycoside. So answer for this 32 is B. Next, which of the following organelle in the figure, they have given some figure there, nucleus, showing nucleus and endoplasmic reticulum, rough endoplasmic reticulum, protein synthesis. B answer is correct. 34th question, the major site for synthesis of lipid. Okay, they are asking major site. 34th, synthesis of the lipid is smooth endoplasmic reticulum. Okay, what it is? Smooth endoplasmic reticulum. Next, the term glycocalyx is used for. The term glycocalyx is used for a layer surrounding the cell wall of bacteria. Okay, so D, that is glycocalyx is nothing but what? It is a cell wall of the bacteria. Next, which of the following types of plastic does not contain stored food material? So, chromoplast, helioplast, alloplast. So, I think 36A is the answer. Okay, where it is not, it is a pigment, it is not showing the storage of the food. Rest, helioplast, alloplast, amyloplast store fat, protein, carbohydrate, food storage. 37. Select the alternative correct identification and function of the organelle A. They are giving what? Mitochondria. That is the mitochondria cellular energy in the form of ATP is the correct. 37A. 38. Select the correct one. Cell membrane. Regarding cell membrane, they are asking. Cell membrane we have discussed about recently. We have discussed now. So tell me which is the right answer. Sodium, potassium. Actually, it should be active. A will not come. Proteins make up 60 to 70 of the cell membrane. Lipids are arranged in bilayer with polar head towards the inner part, that is fluid mosaic model of cell membrane. So D is the correct answer. 38 single and Nicholson. 39th, true about ribosome. 39th, that is ribonucleic acid and proteins are present in the ribosomes. Next, question number 40. So in case of question number 40, so let us discuss about the question number 40. Which one of the following does not differ in E. coli and Chlamydomonas? Okay, which is not differing here, that is in case of 40, cell membrane is not differing. Cellular part correctly described it. 41 I am discussing. 41. So they are thylakoids. First one itself, they are flat, sac-like, already we have discussed. Centrioles, site for active RNA. Ribosomes synthesize RNA. Ribosomes, chloroplasts are larger, while those in the cytoplasm smaller. No. 42. Which one of the following structure is an organelle within an organelle? Okay, they are asking 42, which is that organelle is an organelle within an organelle, that is ribosome. Okay. Peptide synthesis inside the cell takes place in. Okay, where it is taking place? In. Answer is D, ribosome. Important site of formation of glycoprotein and glycolipid in the where? 44th question, that is B. Golgi apparatus, the formation of glycoprotein and glycolipids will occur. Okay, next. Which of the 45th question? Which of the following is not considered as part of endomembrane system? So 45th question answer will be peroxisomes. Okay, peroxisome is not considered to be as endomembrane system. Rest Golgi complex, vacuole, lysosomes, they are considered as endomembrane. So they have given the structure of mitochondria. You have to label the parts. So what will be the answer for 46? 46 will be B. B is correct. Inner membrane forms enfolding called crystal. Next, 47. The plasma membrane consists mainly of proteins, phospholipids bilayer. 47th answer will be B. Next, the main area various activities of the cell takes place, mitochondria. Okay, mitochondria where the various activity will be occurring. 49. Which one of the following has its own DNA? Mitochondria is having its own DNA. So 48th answer will be what? 48th will be cytoplasm. Sorry, C. Which one of following has its own? 49th, yes. 49th answer will be A. 50. Which one of the following structure between two adjacent plasmodesmata? Plasmodesmata, which is connecting from one cell to the another cell. Next, so we have solved nearly 50 questions, and next class we will again discuss about the half part of a cell, the unit of life. We have discussed half part, and rest what all we have discussed here. We have discussed about the cell theory, prokaryotes, eukaryotes, okay. Then cell wall, cell membrane, we have discussed it. Next, one more Neet class we will get after some 1 or 2 weeks. We will continue this topic once again. Okay, so we have to study the synopsis of the cell organelles like plastics, mitochondria, okay, Golgi body, what their structure, function you have to remember, which especially NCRT you have to go through. Then only it will be very easy for you people. Okay. So if you have any type of doubts, you can clarify or you can share that question in your WhatsApp group. Okay. So you can share that. Thank you, students.